Hello, and welcome to this Human Physiology Lab, Biology 206, lab number 7 overview for blood pressure. As always, in each one of these lectures, we have a series of objectives that you can take a look at, and these are posted on the handouts as well. Now, keep in mind that what we're going to be looking at in terms of blood pressure are going to be what's going to be able to be measured kind of out in the periphery. So what we can measure out in the arteries, either during ventricular contraction, corresponding to systole, or during the relaxation and refilling of the heart during diastole. Keep in mind that when we're looking at this, basically during contraction, the ventricles are going to squeeze, the cardiac muscle cells are going to contract, we're going to squeeze the blood and ejected blood out into the aorta. So we can see the aortic pressure kind of increasing during that process of contraction. And then when we go into diastole here, we're going to have kind of the heart going into a relaxation phase. And what we're looking at with the ventricular pressure here in red is going to be dropping down as the ventricles start to kind of relax and refill. But we're going to be focused on the periphery. And so we're not going to measure the aorta directly. But what we're going to see is that during systole, the contraction of the heart is going to eject a bolus of blood out into our arterial system, out into our aorta and our major arteries. Those arteries are going to have smooth muscle within their walls. They're going to have a lot of elastic fibers within their walls. So they're going to be able to expand with this increased volume of blood that has been ejected from the heart. And then when that heart goes into its relaxation phase during that filling phase over here, what's going to happen is that pressure is going to end. The blood in that arterial system is going to want to try to flow back as the ventricles are, in essence, dropping in pressure. They're going to run into our aortic valve right here. So we can hear the aortic valve slam shut at this point. And then we're going to have enough pressure within the system that instead of going back towards the heart, it's going to be carried out again towards that lower pressure out within the periphery. And so if we take a look at that, the overall blood pressure then is gradually going to decrease as we are kind of distributing the blood throughout the body, ultimately returning to the heart at a much lower pressure, kind of over here at the start of our cardiac cycle. So we're going to be focusing in on what's going on in the arteries, kind of out in the periphery during periods of systole and diastole. Now, it's not part of the assignment, but you are going to have a stethoscope and a blood pressure cuff. You have the opportunity to try to listen for the heart sounds. Again, the first sound, the lub sound associated with the ventricles starting to contract, blood kind of flowing back towards the atria, causing our AV valves to slam shut, our atrioventricular valves. And then at the end of systole over here, we're going to have the second heart sound occurring when the blood, again, the pressure in the arteries, the aorta are going to try to push the blood back into the heart. And we're going to cause that aortic semilunar valve to close at that point, giving us the second heart sound. So again, it's not required in this assignment, but we've got the tools necessary. So if you're interested in looking at that. The focus of this week's lab is taking blood pressure, and because we can't say blood pressure, just blood pressure in science, we're going to refer to it as auscultation. And so basically the idea is that if blood is flowing normally through our arteries, it's going to be flowing smoothly. There's not going to be a sound associated with it. As we increase the pressure within our blood pressure cuff, that pressure in the cuff is going to be squeezing the arm, and basically it's going to exceed the amount of pressure within the arteries, and it's going to cause the artery to collapse. Again, what we're looking at here is going to be the relationship between the pressure inside of the artery and the pressure outside of it within the blood pressure cuff. Now, when the artery is completely collapsed in this situation, the blood pressure cuff is at a high pressure. We're squeezing it completely so that when we are listening for a sound kind of below or kind of peripheral or more distal to where the blood pressure cuff is going to be located, we're not going to hear any sound because there's no blood flowing through the blood vessels or arteries at that period of time. As we go over here to that range between the systolic, when the heart is contracting, and the diastolic pressure when the heart is relaxing, what we're going to have is during heart contraction, we're going to increase the pressure within our arterial system, increase the pressure pushing back against our blood pressure cuff, and we're going to be able to squeeze through a little bit of blood at those higher systolic pressures during the cardiac muscle contraction, during the heart contraction. So what we're going to hear are little kind of bursts of sound that are going to be called the Korsakoff sounds. And this is basically going to be kind of turbulent flow of blood kind of coming through these arteries. And so it's not coming through smoothly. It's almost like it's running over a babbling brook. And so, the stream is going and it's kind of cascading over a series of rocks and it's generating a sound. In this case, the blood is kind of flowing through a partially collapsed artery because the pressure within the cuff is exceeding the pressure within the blood vessel. Ultimately, when we get down here to the diastolic pressure and lower, we are going to have the situation where the pressure within the arteries is going to be higher than the pressure within the blood pressure cuff. And at that point, the blood pressure cuff isn't going to be able to squeeze the artery at all, again, because the pressure is higher in the artery than in the cuff. And so we're going to have, again, smooth blood flow through that area. And so it's going to be quiet, again, similar to what we had over here with the normal blood flow, because we haven't impacted on the wall of it. We haven't caused any, partial occlusion or anything going on. We're not doing anything to contribute to the babbling brook. We're not doing anything that's going to contribute to the turbulence that we can hear with our stethoscope. Now, as I mentioned, the Korsakoff sounds are what we're listening to when we're hearing the blood pressure. So on this one, what we've got on this graph, we've got along the y-axis blood pressure. And so kind of 60 milligrams of millimeters of mercury here, 140 up here. So 140 kind of higher than our, traditional way of looking at the blood pressure, the traditional way of describing a normal, or at least stereotypically normal. I'm not sure what normal means when we start looking at some of these medical measurements, but it's usually described as 120 over 80. And so if we are at a pressure higher than 120, this is going to be that line that is going down through here, kind of diagonally across this graph. This is going to be cuff pressure. When the cuff pressure is higher than our diastolic pressure, basically what's going to happen is the blood pressure cuff is going to completely squeeze off our artery, and it's going to block the flow of blood. And so what's going to happen? Our arterial pressure is going to be kind of oscillating between kind of a higher pressure during our systole, dropping during periods of diastole, the systole, the contraction of the heart, diastole, the relaxation of the heart. So as we approach, our cuff pressure approaches our diastolic pressure, this is where we're going to have these blood vessels kind of partially open in terms of pushing against the wall of the blood pressure cuff, kind of exceeding the pressure that the cuff is going to be exerting on the blood vessel. And we get a little squirt of blood kind of squeezing through the blood vessel at that point, a little bit of turbulent flow and a little bit of the Korsakoff sounds. Now, these sounds are gradually going to get larger as our cuff pressure continues to drop. We are going to have kind of larger bursts of blood, larger volumes of blood that are going to squeeze through with each contraction of the heart. But ultimately, it's going to drop off, because as that diastole is going on, we're going to see a decrease in the pressure. And when that pressure drops down, the pressure within our arteries drops down below the blood pressure, cuff pressure, we are going to have that artery going to be occluded. Ultimately, when we get down all the way over here to where the Korsakoff sounds disappear, what we're going to have is that the pressure within our arteries is going to remain higher than our cuff pressure. And so it is going to prevent the blood pressure cuff from causing, a partial or complete occlusion, just because the arteries maintain a higher level of pressure, higher pressure than what we're able to exert with our blood pressure cuff. Now, what is it that we're actually going to be doing in lab this week? So again, same basic format that we've been doing throughout the course. All of the materials are going to be available on Canvas. We're asking you to download the lab report template. So again, that's available on Canvas as a Google Doc file. You can save that onto your Google Drive if you want to use whatever the Google Doc program is to edit it there, or you could download it to your computer if you're going to use like a Microsoft word processing program. Again, asking you to use the naming convention so that if we have to download these, we don't have to rename them and try to figure out, who it is that submitted that report. Now, the first activity is going to be conducted without the Power Lab system and lab chart. And so you're going to have the blood pressure cuff, a stethoscope, and you're going to take blood pressures of other members within your lab group. And I encourage you to kind of spread these responsibilities around so that everybody gets the opportunity to take a blood pressure. We've included a video, a relatively short video on how to take a blood pressure manually, which again will provide you real key insight into doing that. And so you're going to take some individual blood pressures and then record them in the table. In the second activity, we're going to start to use the lab chart system in the Power Lab. And we're basically going to be measuring the pulse pressure and pulse recordings by taking a look at the arterial pressure during systole and diastole. So again, follow the instructions within the ADI handout. They provide a lot more detailed instructions than the lab report handout, the lab report template. The third activity, focusing in on blood pressure and arterial sounds. So a similar activity, so using the blood pressure cuff on the pulse transducer, but we're also going to be using a cardiophone. And so this is a substitute for the stethoscope, but it goes along with being able to collect the information within our Power Lab systems. And then the final activity in this week's lab is going to be looking at the effect of the cuff position, or basically looking at the effect of a drop in arterial blood pressure as we get further and further away from the heart. So the way we're going to do that is we're going to take the blood pressure cuff, take it in our traditional location, taking the blood pressure on the brachial artery, but then we're also going to put it onto the wrist to take it on the radial artery, and then the ankle, as well as the thigh, to take different measurements out in the periphery, different arterial pressures in the periphery of the body. As always, we're asking you to submit your lab report. And again, we've got that on Canvas. Submit lab report number 7 here. And if you've got some good data files that are labeled, we're again trying to collect these so that if we need to, we have resources available that students can complete this lab remotely. And we've got a variety of data that will be available to them. So it's optional. You don't have to do that. But if you have good data sets, I'd really appreciate it if you could share those with me. As always, your lab instructor may have additional information available to you. And so please see if they have any changes or updates for the lab for this week. As always, if you have any questions, please do not hesitate to reach out to me. Thank you, and have a great week in lab.